How much water do you think it takes to grow one dry 40-pound bag of alfalfa? If you thought the correct answer was number 4, you were correct. A tremendous amount of water. Leaves have numerous openings called stomata. When the stomata are open, water evaporates from the interior of the leaf to the outside atmosphere in a process we call transpiration. This water loss, of course, has to be replenished as movement of water from the soil through the plant and to the atmosphere along an interconnected film of liquid water. The tension created by the evaporation from the leaf draws water up the xylem vessels because water forms hydrogen bonds. And this is what produces the unbroken interconnected column of water. The water movement through what we call the soil-plant-atmosphere continuum is driven by the passive movement of water generated by that gradient in water potential energy. The energy is created by a difference in water potential from high potential in the soil to a gradually more negative potential in the atmosphere. A stronger rate of evaporation will lead to more tension in the water column and a faster rate of water movement as long as soil water is not moving. We may look at this process as a flux of water in response to a driving gradient moderated by a conductance or resistance to that water movement. When soil water is limiting, the unbroken column of water may indeed be broken. We call this cavitation. The cohesive water column then breaks up and air disconnects the continuous pathways of water from the soil to the leaf. And the tension at which this happens is approximately the tension of the permanent wilting point. The soil-plant-atmosphere continuum is fundamental to the hydrologic cycle and, of course, to the ability of plants to photosynthesize. So transpiration is important for photosynthesis, for a plant to capture carbon from the atmosphere, combine it with water to produce glucose and other sugars. However, there's only 4 CO2s in the atmosphere for every 10,000 water molecules, meaning that if a plant keeps its stomates open, it will lose a tremendous amount of water in an effort to capture some CO2. The end result being that only about 1 and maybe up to 5% of the water that is taken up by the plant and moved into the atmosphere is used for photosynthesis. So what's the rest of the water being used for, and is it a beneficial use for that water? Well, some of this water helps transport nutrients to the plant roots. So the water flux from the soil to the root supports the transport of nutrients. It helps uphold turgor pressure, which is a structural requirement for the plant, and it also serves in the cooling of the canopy. Remember, evaporating water requires lots of energy, and that will cool the canopy. And there are also a whole bunch of other uses for it. So it is not entirely a complete loss of water. So how is this water loss controlled? Well, there's an atmospheric control, meaning we need to have adequate energy to evaporate water. We need to have a mechanism to remove the evaporated water for continued evaporation. So the atmosphere controls that water loss to a great degree. The plant also has a control on it through its stomatal opening. It can, in response to environmental stimuli, open or close its stomates, thereby reduce the amount of water loss. Of course, that reduction in water loss comes at the expense of not having the stomates open to be able to take in CO2. And the third control is the control that the root zone has on that water loss. Here, if we want to envision an elongated root for a moment, that root is surrounded by soil that is directly influenced by what is happening with the root and the root water uptake, and of course the bulk soil. Any water being lost from the leaf has to be replenished through root water uptake, and bulk water has to move from the bulk soil through that soil surrounding the root into the root. Of course, as the plant is doing that, as root uptake is taking place, we will develop a potential gradient between the root and the soil, which is going to limit the uptake of water. So as we reduce the water content in the soil surrounding the root, we also reduce the hydraulic conductivity of the soil water in that soil surrounding the root. We also have less contact points between the root and the soil surrounding it as we desiccate and shrink that soil volume. The end result being is that the flux from the bulk soil to the root reduces with time. But with all the interest on controlling transpiration, let's not forget that yield and transpiration are really closely related with each other. So look at the following graph where we look at yield versus the annual water use. And you see we need a little bit of annual water use to get any yield started. But once we apply water, we get an almost linear relationship between yield and transpiration. If we redefine water use slightly and we look at water use in terms of actual water applied, meaning we have some losses in terms of evaporation, runoff, and drainage, then we get a slightly different curve, which shows that there is a production limit, a maximum yield we can attain with water. And of course, it also implies that there is an optimum water use yield efficiency that can guide our irrigation decisions. So how do we make these irrigation decisions? One way is to measure evaporation from a water pan as a standardized open water surface from which we can measure the amount of water lost due to evaporation over periods of hours, days, and weeks. The other one is to use what's called a reference surface, which usually is a well-watered turf grass that serves as a standardized surface to estimate evapotranspiration from. These two can be made equivalent to each other if we multiply the water pan evaporation by a pan coefficient to gain what is somewhat of a potential evapotranspiration rate, or ET0. It's called a potential rate because it really only accounts due to water loss in response to atmospheric conditions, to the temperature, radiation, wind conditions. So in other words, how much energy was available to evaporate water and what were the atmospheric conditions that contributed to removal of water vapor. It does not account for differences in plants, nor does it account for limitations of water availability in the soil. In order to do this, we multiply the potential evapotranspiration rate with a crop coefficient and a crop stress coefficient to gain the actual estimated evapotranspiration rate. The crop coefficient accounts for differences in crops. An alfalfa crop is going to behave different than a corn crop, which is going to behave differently than an almond orchard. But it also accounts for differences in life stage for where transpiration increases as we have more full canopy closure as well as root development with life stages. The stress coefficient accounts for differences due to salt stress, and it accounts for limited soil water availability. The limited soil water availability can be estimated with what's called a Fettus root water uptake function, which relates the uptake of water to the matrix potential and has a plateau in which water uptake by plants is not limited by soil water conditions, meaning there is sufficient soil water available to not limit the root water uptake. But to the left of that, if we are to the left of the field capacity water content, we know that too much water will limit the water uptake. And also as we move towards the permanent wilting point, as we get to drier and drier soil conditions, soil water is more and more limited, and the uptake rate by a particular crop will be limited then. So we take all this together, we have estimated an actual evapotranspiration rate that then we can use to make smart irrigation decisions, to where we know exactly how much water we need to put back in to replenish what the crop has transpired into the atmosphere.